Good afternoon, students, and welcome to today's NPTEL problem solving session. We'll be solving week 11 lecture problem set for today, and we'll begin shortly at 3:05. I'll keep admitting students meanwhile they are requesting to join this call, and we can then start at 3:05. And good afternoon to you all. For all those who haven't joined this session, can view this lecture recording on YouTube. We'll have one more week to go after finishing week 11th, and then we can solve all the practice problem set corresponding to the lectures in NPTEL. So we'll start shortly at 3:05 and hoping to see solve questions with you. Yes, Ajay, you have to tell me something. Okay, okay, we'll start soon. Yeah, so it's 3:05 currently, and I'll start sharing my screen. So we'll follow the same format. We'll have 10 exemplar problems corresponding to lecture week 11, where Professor Subramaniam is teaching us about mammalian development, and we'll take some questions similar to what we receive in the quizzes and assignment and try to solve them. Let me present my screen. I hope all of you can see my screen and hear me properly. Let me just switch on my laser pointer and let me know in the chat box with a yes or no. Then we'll move forward with the questions. And to the new students who have joined, please turn off your mic and video chat for smooth conduction of this session. You can communicate with me via the chat box, and if you have any doubts or you want to speak to me, I'll give you a chance. So do not worry. Hello, hello, hello. Sorry, students, for the delay. I think I lost my connection. I'll try resharing my slides and we'll begin the session from this beginning again. And to all the students attending this call, please turn off your mic and video chat for smooth conduction of this session. Yeah, I hope you are now able to clearly see my presentation and also the slides changing. Are you able to hear me properly, the students? Please let me know with a yes or no from the chat box. Then we can proceed with the questions. Uzma, can you confirm? I think you are the student here. Are my slides visible to you and can you see them changing? Anyways, let me start the presentation. So, I welcome you all to problem solving session for Introduction to Developmental Biology of NPTL. The course instructor for this course is Professor Subramaniam, and my name is Ashutosh. I'm a PhD scholar at IIT Kanpur. I'll be the teaching assistant for this course. So, we have gradually moved from problem solving session week 1 to week 11, and now we are one problem solving session short for completing this whole course. And what we'll do today, as I lay down the rules, since we have been doing this practice over the last problem solving sessions, I'll post 10 questions similar to what you receive in your quizzes and assignments. These are multiple choice questions. I'll post the questions and the students have to find out the correct answer or guess the correct answer. Then after collecting some of the correct answer, I'll reveal what is the actual correct answer and we'll understand the rationale and concepts behind the correct answer. So that's how we'll proceed with this session. Moving on to the first question. So this problem solving set will be corresponding to lecture week 11, where Professor Subramaniam is teaching us about early mammalian embryo development and EVO-DEVO, evolutionary developmental biology. So the first question is on your screen. Which of the following is true? So as I said, these are multiple choice questions. So there may be one or more than one options correct. You have to tell me which of the following is are true. Option A, the umbilical cord has blood vessels that directly interconnect the blood vessels of the embryo and the mother. Option B, a chimeric female mouse that displays two different codominant coat colors will give rise to progeny with a similar mixed coat when mated with a colorless or white male. The third option, precaudal mesoderm is formed from the node. And fourth option, Fox genes specify the anterior posterior axis up to the forebrain. So these are the four options before you, and please mention in the chat box which of these do you think is true. Option A, B, C, D. So let's wait for a minute to collect some answers. So we have two students with us, Ajay and Uzma. So I expect both of you to answer. Ajay has already answered. Uzma, what do you think is the correct answer to this question? It's okay if your answer is not correct. It's fine. Do not feel shy. You can just guess based on your logic or anything you can remember from the lectures. Anyways, we'll move forward with the collected answers. So Ajay has answered option 3, and option 3 is the correct answer. Precaudal mesoderm is formed from the node. So this is the true statements among all the four options. So the first option, umbilical cord has blood vessels directly interconnecting the blood vessels of embryo and mother is not a true statement, and a chimeric female mouse with two different codominant coat colors will give rise to progeny with a similar mixed coat when mated with a colorless white male. This is also not true, and Hox genes specify anterior posterior axis from the whole body plan, not just up to the forehead. So the whole anterior posterior body plan of the embryo is uh, delineated by the Hox genes. So let us have a brief look about the the umbilical cord. What is the umbilical cord, and what actually serves as an interface between the fetus and the mother? So extraembryonic tissues are those tissues which are formed from the trophoblast. If you remember, the inner cell mass and the trophoblast in the blastocyst stage, the inner cell mass constitute of totipotent cells which can give rise to the whole embryo, and the trophoblast is responsible for extraembryonic tissues. So the placenta is such a structure which arises from the trophoblast. Placenta is a villus or a interconnection between the developing embryo or fetus, and the mother during pregnancy. So we have tissues from the mother and tissues from the fetus forming the interconnection of the placenta. Placenta is an specialized organ that develops within the mother's uterus and facilitates exchange of nutrients, oxygen, and waste products between the maternal bloodstream and the fetal bloodstream. So, since a fetus is an independently growing organism within the mother, we have a separate blood bloodstream for the fetus and a separate bloodstream from mother, and these get interconnected within the placenta, but not the umbilical cord. While the umbilical cord constitutes of the fetal arteries and veins. So umbilical cord consists of two UCB, HSPC, MSC, MSC. So you see here, this is a chorionic plate. This is a umbilical cord. Umbilical cord connects the placenta to the fetus. We have the chorion, and this decidua basalis and villi. These form the placenta. So the first option, umbilical cord has blood vessels that directly interconnect the blood vessels of both the embryo and mother, is not true. The correct statement is the placenta has blood vessels that directly interconnect vessels of the embryo and the mother. To the second option that we faced here, a chimera with two different codominant coat colors. So let's say this is an example of a chimera with a brown coat color and a black coat color when crossed with a white mouse, which is colorless. Now what would happen? What will be the progeny? If we assume that two codominant coat color genes in the female mouse are A and B, let's say this is a female mouse with two codominant coat color genes, gene A and gene B, and the white coat color gene is W. Now if A and B are codominant genes and expressed in a mixed coat color, since it is a chimera, some cells will be having expression of A, some cells will be having expression of B, and some cells will both be having expression of A and B. So it depends. It depends on what kind of cells form the germline in a chimera. So the germline constituent cells will produce a gamete having the same genetic constituent or expression pattern of the parent, and then when we mate such a chimera with a wild type mouse, then what can be the possible outcomes? So first outcome would be if both the codominant genes A and B are codominantly expressed in the offspring. So the offspring can have either sole expression of A, sole expression of B, or both expression of A and B. And since white color is a uh, wild type allele, so this chimeric coat color will be dominant over the white colored. So the offspring having both the genes expression A and B will have similar mixed coat color as its female. So this is one possibility, but this is not the total possibility. So there will be offsprings if one of the codominant genes A or B is expressed dominantly over the other and the white gene W is recessive, then some of the offsprings may have the mixed coat color, others may have white coat color depending on which genes are inherited. And if both codominant genes A and B are recessive to the white gene, so this is also a relationship. So we know that A and B are codominant, but they may not be dominant over the white gene. So if A, B are both codominant but are recessive to the white gene, then the offspring may predominantly have a white coat color. So there are possibilities. And the specific outcome will depend on genetic makeup of the codominant genes and their interaction with the white coat color gene in the progeny. So there are tons of possibilities that we can deduce from a chimera with two different codominant coat colors when crossed with a wild type mouse. So statement 2 will also be not true because one of the possibilities is similar mixed coat. Other possibilities are white males uh, or either of the color of the two codominant coat colors. And as I mentioned in the question, Hox genes specify the anterior posterior axis from forebrain starting to the tip of the last vertebra. And the third option, which was the correct option, the precaudal mesoderm arises from the node. So if we look at the chicken embryonic development, precaudal mesoderm, also known as the head mesoderm. So precaudal mesoderm is the head mesoderm, is a region of mesodermal tissue that forms in the developing embryo. So it's a kind of mesoderm as its name suggests, and it forms the head. And through the node, which is also known as Henson's node, it is associated with the primitive node, which is a structure found in early stage of embryonic development. Precaudal mesodome is specified from cells within or near the primitive node. So these cells undergo migration. These cells move inside from the primitive node along the length forming the primitive streak. So there are always cells migrating inside and rearranging themselves. The mesodermal tissue is important for the formation of head and cranial structures in the developing embryo. It gives rise to various cell types and tissues that contribute to the head region. So some of the cells, these mesodermal cells travel inside, migrate inside the Henson's node. Migrating cells are these mesoderm, and this will form the precaudal mesoderm. So precaudal mesoderm forms from the node. Third statement was the correct statement. Precaudal mesoderm is formed from the node. Good. Ajay answered is very correctly, and that is very good. We'll quickly move on to the next question. Which of the following antagonize BMP at the anterior end of the embryo? If you remember BMP antagonizers or inhibitors, which are important players in the anterior end of the embryo. Option A, cordon. Option B, Wnt. Option C, FGF8. Option D, noggin. Option E, lefty1. Option F, nodal. So there are six options, and there can be multiple answers correct. You have to tell me which of the following antagonize BMP at the anterior of the embryo. Let's take a minute to collect some answers. I see Ajay has already answered cordon and noggin. Uzma, are you able to follow the question? Will you be able to answer? Which are the BMP antagonists present at the anterior side of the embryo? It's okay if you don't know the exact answer, you can guess based on logic or anything that you remember from the lectures. Okay, we'll move forward. So I'll reveal the correct answers now. Which of the following antagonize BMP at the anterior end of the embryo? First correct answer is cordon. Cordon is a BMP inhibitor, specifies the anterior side of the embryo. Second correct answer is noggin. So cordon and noggin are the correct answers. And Ajay has answered very correctly. Very good. Good. So if you follow the schematic here, noggin, cordon, follistatin, these are some of the important inhibitors of BMP which specify the future anterior region. So here is an example of frog embryo, developing embryo, where we have the animal side and the vegetal side, and the orange portion here is where noggin, cordon, follistatin start expressing, where they inhibit the expression of BMP4 or 27. And so thus the BMP inhibition is necessary for specifying the organizer as well as the anterior end present in the anterior end of the embryo. It's also important. So BMP is important for inducing neural fate to epidermal fate. So the epidermis neural structures will form here, and the rest of that will form the epidermis. And noggin, cordon, follistatin are the important inhibitors. BMP inhibitors such as noggin, cordon play critical role in regulating BMP, binding to BMP molecules, preventing them from activating the receptors. So these are such inhibitors which will bind to the ligands of BMP signaling and prevent them from binding to the receptors. By inhibiting BMP signaling, these molecules help establish anterior side of the embryo, allowing anterior structures to develop in the absence of strong BMP mediated posteriorizing signals. So BMP signals are posteriorizing signals. It means wherever there is high BMP expression, that part of the embryo is going to be the future posterior side. So wherever we have BMP inhibitors will be specified as the anterior side. The presence of BMP inhibitors in the future anterior side of the embryo is crucial for the proper patterning and development of anterior structures such as head and brain. So the precaudal mesoderm which will form the head structures and the those neural fate of the neural tube forming the brain will be specified by the presence of these BMP inhibitors, noggin, cordon, and follistatin. These inhibitors help counteract posteriorizing effects of BMP signaling, allowing anterior structures to develop and differentiate appropriately. So wherever we have high BMP, that will be the future posterior side. Where we have inhibition of BMP, that will be the future anterior side. And Ajay have answered very correctly, and we have very good. Okay, moving on to the next question. Question number 3. Hox A6 in vertebrates is a dash of Antennapediae in Drosophila and a dash of Hox C6 in the vertebrate. And your options are 1. Ortholog, paralog, second, paralog, ortholog, third, both are orthologs, fourth, both are paralogs. So you might have to go through the question quickly once more. Hox A6 in vertebrates is a dash of Antennapediae in Drosophila and a dash of Hox C6 in vertebrates. So we have one gene in vertebrate comparing it with another gene in Drosophila and another different gene in vertebrate. You have to tell me which of these is an ortholog or paralog, or they are both orthologs or both paralogs. Let's wait for a minute to collect some answers. Okay, I can see some answers. Ajay had answered option 1. Hox A6 in vertebrates is an ortholog of Antennapediae in Drosophila and a paralog of Hox C6 in vertebrate. Let us check if this is the correct answer. Absolutely correct. Hox A6 is actually an ortholog of Antennapediae and a paralog of Hox C6 in the vertebrate. So let us understand what are orthologs and paralogs. So to be straight in simple terms, paralogs are genes that result from duplication events within the same species, while orthologs are genes in different species that share common ancestral gene due to speciation. So paralogs are genes which are duplication events. Duplication event means those two genes might be different because in during evolutionary time point the gene got duplicated, acquired some mutation, and now the gene is somewhat different from its ancestral gene. So such genes are called paralogs within the same species arising from duplication events, while orthologs are genes in different species that share common ancestral gene due to speciation. So orthologs are similar genes present in two different species. Paralogs are different genes present in the same species. Paralogs are found within the same species, can be present on different chromosomes or within the same chromosome. They often have similar functions or may have diverged in function after duplication. So this is what was a possibility I was talking about. A gene has copied itself. Now it can be preserving the similar function, or can diverge in function after duplication. Paralogs can contribute to gene family expansions and functional diversity within a species. So, functional diversity within a species is brought about by paralogs, whereas orthologs are genes found in different species that share a common ancestry due to speciation. So, two different species and the same genes, those are orthologs. Same species, two different genes, those are paralogs. Orthologs arise from the same gene in the last common ancestor of the two species. Orthologs typically maintain similar functions or roles across different species because they have been inherited from the common ancestor. So generally we assume that orthologs are the genes having similar function but in two different species. Here is an example on the right, globin gene. Globin gene is the best example to understand what are orthologs and paralogs. So there was a gene duplication event in globin gene, and each globin gene has the alpha chain and the beta chain. So in the alpha chain, we have frog alpha, human alpha, and mouse alpha. Human alpha and mouse alpha share much more similarity than the frog, so they appear later in the phylogenetic tree. So if you compare frog alpha globin gene and mouse alpha globin gene, these are orthologs because they are from two different species and the same kind of gene that we are talking about. Now similarly, the beta chain gene also has its variants: mouse beta, human beta, and frog beta. If we compare mouse alpha and mouse beta in the same species, they are two different genes, alpha chain and beta chain of the globin family. These genes will now be called paralogs. Within the same species, two different genes are called paralogs. Same genes within two different species are called orthologs. I hope this is clear to the students. And here is another phylogenetic tree showing us the modes in which orthologs and paralogs are generated. We have duplication leading to gene duplication, mostly leading to paralogs, because duplication and then mutation or acquiring new properties will lead will render one gene different from the other in the same species. Speciation node, where the common ancestor lays down the same gene or same protein plan into two different organisms. So ortholog, so HSAP is humans, MMUS is mouse. So we can see humans and mouse comparison is an ortholog. Between species paralog, within species paralog is what we were mostly referring to. These blue ones, within human and human we have two different genes. Orthologs again with human and mouse, and within species paralog within mouse and mouse. So this is a complex example showing the difference between an ortholog and an paralog, and Ajay has answered correctly. Very good. Moving on to the next question. Knockout of all alleles of HOX6 will result in which of the following? Knockout of all alleles of HOX6. Option A, the thoracic vertebra T1 will now show lumbar phenotype. Option B, the thoracic vertebra T1 will now show cervical phenotype. Option D, the thoracic vertebra T1 will form ribs. Option D, no change will be observed. So if you remember, if you remember the specification expression of Hox gene patterning the whole vertebra in vertebrates, and there are different types of vertebra. We have thoracic vertebra, we have cervical, we have lumbar, and we have caudal, we have sacral. So all different kinds of vertebra. You have to remember where is the expression pat, where is the expression of Hox6 along the vertebral column. Then you will be able to answer this. If all alleles of HOX6, all alleles of HOX6 are knocked out, what will be the resultant phenotype? Okay, I can see some answers. Thoracic cervicalization option 2. Okay, so Ajay is answering option 2. Thoracic vertebra T1 will now show cervical phenotype. Let us check out what is the correct answer. Ajay is absolutely right again. Thoracic vertebra T1 will now show cervical phenotype due to knockout of HOX6 gene. So here is a schematic showing the different patterning HOX genes, homeobox genes along the vertebral column. So we have the whole vertebral column is divided into different regions. So we have first 7 vertebra, the cervical vertebra or the neck head forming vertebra, 13 thoracic vertebra, thoracic or the ribs forming, 6 lumbar vertebra, 4 sacral and 20 caudal. So this is the arrangement of vertebrae in vertebrates, and we have different gene clusters, homeobox genes cluster expressing along the length of the vertebra. HOX4 and HOX5 specifying the cervical vertebra, HOX6 specifying HOX6 and HOX9 specifying thoracic vertebra with initial segments specified both by expression of HOX5 and HOX6. Overlapping expression of HOX10 uh, specifies lumbar vertebra, and HOX11 overlapping HOX10 specifies sacral vertebra. So these are different HOX genes that we were talking about. So there are three different genes of HOX11, three or two different types of HOX6, HOX9. So we have this gene cluster expressing in the thoracic. The color coding, if you follow, 7 cervical vertebra is specified by HOX4 and 5 here, and in the somites where we have this green expression HOX1, 2, 3, and HOX10 specifies lumbar vertebra. Now what will happen if we mutate or you knock out all alleles of HOX6? Let's say for 6 we knock out HOXA6, HOX6A, HOX6B and HOX6C. Then what would happen? So here is another nomenclature of naming the vertebra. The first vertebra of the thoracic segment is called T1. It's very simple to understand. T for thoracic and first segment. So the first segment of thoracic, if you see, is specified by HOX6 and as an expression of HOX5. Now what happens if we knock out all the alleles of HOX6? Then the HOX5, the vertebra specified by both expression of HOX5 and HOX6 now does not see any HOX6. So it will act as uh, HOX5 expressing vertebra or the cervical vertebra, which will, so T1 will be looking like neck vertebra or the cervical vertebra. Knocking out HOX6 genes may lead to homeotic transformation. So such transformations are called homeotic transformation, where the identity of one or more thoracic vertebra becomes more similar to adjacent segments. So here we saw knocking down of HOX6 in T1, T1 which is specified by both expression of HOX5 and 6. Now that 6 is gone, HOX5 expressing segment will now look like cervical. For example, without HOX6 function, one or more thoracic vertebra may take on characteristics more typical of cervical or neck vertebra. So this was the correct answer, and students have answered very correctly. Very good. Okay, moving on to the next question. Question number 5. Division at which time point in embryonic development gives rise to twins developing it with one amnion but two chorions? I'll repeat the question again. Division at which time point in embryonic development gives rise to twins developing with one amnion but two chorions? We have one amnion and two chorions. Option A, at the morula stage. Option B, after the trophoblast is formed. Option C, after amniotic ectoderm is formed. After option D, After primitive groove is formed, option E, none of the above. So you have to mention the stage in which if we uh, divide the embryo, it can give rise to twins developing with one amnion but two chorions. Let's take some time to answer this question, and I'll reveal I'll reveal the correct answer. So if you remember, Professor Subramaniam in his lectures week 11 at NPTL have discussed a similar kind of case for identical twins, monozygotic identical twins, and double fertilization fraternal twins. If you remember those slides, or uh, if you have learned a bit about the development of chorion and amnion, you might be able to answer this. Division at which time point in embryonic development gives rise to twins developing with one amnion but two chorions. I see Ajay has answered not possible, none of the above. Let me quickly reveal the correct answer to this question. Again, Ajay has answered very correctly. None of the above. Such an embryo is not possible. An embryo with one amnion but two chorions. So you have to understand the difference between amnion and chorion and the point of origin of these two tissues. Then we'll be able to deduce why such an embryo is not possible. So even if you divide the embryo at morula stage, or after the trophoblast is formed, or an amniotic ectoderm is formed, or after the primitive group is formed, none of them could lead to a twin developing with one amnion and two chorions. This is practically not possible. So here is the exact representation which Professor Subramaniam has used in his slides. So here we were talking about twins, monozygotic twins and dizygotic twins. Monozygotic twins result from single fertilization. So one egg fertilized by one sperm, but when the zygote is in two-celled stage, there can be different fates. If due to if due to a chance these two cells got physically separated, then each of them having the same genetic makeup of the initial zygote can now form a blastocyst with two different inner cell masses and two different trophoblasts. So two different individual blastocysts, and two such blastocysts can implant. So we know that amnion is the ICM derived or inner cell mass derived. Amnion is an embryonic structure specific to the fetus. The epiblast will give rise to the amnion. Amnion is derived from the inner cell mass, while the chorion is the outer extraembryonic structure derived from the trophoblast. So now the number of trophoblasts will determine the number of chorions, and the number of ICMs or inner cell mass will determine the number of amnion. So we here will generate two chorions and two amnions. So if these two get implanted in the mother's womb, then we'll have a womb containing two twins with two chorions and two amnions. On the other case, when some some other chance event could happen, that this cell at an 8 cell or 16 cell stage forms two different inner cell masses. Now what but within a single trophoblast. So these two inner cell masses will form two amnions, but the single trophoblast would be forming only one chorion. So here we can have a situation where the twins are with two amnions but in a single chorion. And in the other stage, and in some very rare cases, the trophoblast can be intact, but the inner cell mass have two different constitution. inner cell mass are not very far apart to form two different amnions. So in such cases, we can have one amnion and one chorion, which is also the case for a normal zygotic development. But here we can have two twins because the inner cell mass is a totipotent cell mass, can give rise to all the embryonic structures, but not the extra embryonic tissue, which is only given rise, which is only driven from the trophoblast. So amnion is inner cell mass derived, chorion is trophoblast derived, and the question mentioned one amnion with two chorions. One amnion with two chorions would mean that one inner cell mass with two different trophoblasts, which is practically not possible. That's why none of the above is the correct option. So such a case is not possible. Division at any time point will not give us an embryo with one amnion but two chorions. Good. Ajay has also answered this correctly. Very good. Well done. We are quickly moving ahead to the next question. Question number 6. How can variations arise if development is tightly regulated? So this is a question related to evolutionary developmental biology, EVO. How can variations arise if development is tightly regulated? Option A. Anatomic and DNA modularity allows certain parts of body to change without altering the functions of the other parts. Option B. Variations are not at all tolerated during development. Option C. When gene duplication occurs, one of the copies can accommodate mutations and give rise to variations. Option D. Because of molecular parsimony, small variations in the genetic level lead to dramatic changes in the body plan which are embryonic lethal. So these are the four options. And you have to tell me how can variations arise if the embryonic development is so tightly regulated? What are the uh, what are the what are the entities which bring on the evolutionary changes in an organism? If development had no chance of error and is very tightly regulated, then how is genetic variability brought upon in an organism? Okay, it's time for me to reveal the correct answers. How can variations arise if development is tightly regulated? The first correct answer is option 1. Anatomic and DNA modularity allows certain parts of body to change without altering the function of other parts. So due to modularity of the DNA, certain specific parts of the body can change without altering the functions of the other parts. And the second correct answer to this question is option 3. When gene duplication occurs, one of the copies can accommodate mutations and give rise to variations. So this is the second correct answer. Gene duplication occurs, then one copy can stay the same and the other copy can accommodate mutations, give rise to a new function and variation. So let us understand these two concepts for variability in development. The first concept was DNA modularity. DNA modularity refers to the concept that DNA, the genetic material in living organisms, is organized into distinct functional units or modules. So DNA as a whole is organized into distinct units, and at a given particular time only one unit of it is activated, manipulated or repressed without affecting other units. These modules are responsible for carrying out specific biological functions, can be thought of as building blocks of genetic information. So many such modules combine together to carry out a biological function, and these are building blocks of genetic information. Alterations in one module exist independently of the other. So this is what I was talking about. If you alter one such DNA module, then another DNA module which is independent of this particular DNA module is unaffected. So that's how the DNA modularity brings about variation in changes. Here is an example schematic showing expression of different genes in mouse tissues. Let's say gene A is expressed in the brain as well as in the limb. So what does it mean? For gene A, we have two different enhancers. Which this is an example of enhancer modularity, which is a regulatory region regulating the expression of downstream genes. So we have a brain specific enhancer and a limb specific enhancer, which are both turned on, which gives us the expression of this gene in brain as well as in the limb. mRNA expressed in brain and limb when we have two different enhancers. Now, there are proteins, there are transcription factors and mediator complexes which bind these specific enhancers. They have specific proteins. So, in the brain tissue, only a brain specific transcription factor can bind this brain specific enhancer, recruiting RNA polymerase II, the promoter of gene A, and causing expression of gene A. Thereby, whenever we see the enhancer sees the expression of brain expressed transcription factors, it will express this gene A. Similarly, in limb, there is no brain specific transcription factor, rather, there are limb specific transcription factors which can recognize their cognate enhancers and then lead to expression of downstream gene. So we have two different enhancers controlling the expression of same module, same gene expression in two different tissues. So this is enhancer modularity is a is a working model in how DNA modularity without any mutation or changes in the gene itself, DNA expression can be differential, and this leads to variation. One of these leads to variation in evolutionary biology. Similarly, gene duplication and divergence is another method. Gene duplication and divergence is a contribution to evolution. Duplication allows formation of redundant structures, and divergence allows these structures to assume different roles. So in duplication, we have the exact same copy of the gene or any structure, and then these copies can acquire different mutations upon time and can diverge from the original copy, thus undergoing divergence. So duplication allows formation of redundant structures while divergence allows structures to assume different roles. And what is molecular parsimony? Molecular parsimony says that although development differs enormously from lineage to lineage, development within all lineages use the same type of molecules. So we have the same type of toolkit for all the organisms and body plans and any tissue that that will developmentally originate. This is known as molecular parsimony. The simplest toolkit required for development of different enormous structures from lineages to lineage. The transcription factors, paracrine factors, cell adhesion molecules, signal transduction cascades are remarkably similar between different phyla. So this is an example of molecular parsimony in which we have same set of transcription factors, paracrine factors, and different other proteins, which are the minimum requirement of a toolkit that that is necessary for development of an organism. So these two options are correct. The first option was DNA modularity, where independently change in one module will not affect the other module, and the second correct option was uh, gene duplication and divergence. These two phenomena are known for bringing out variations even when development is tightly regulated. I hope this is clear to the students. Please let me know with a yes or no in the chat box. Okay, good, cool, very good. Let's move on to the next question. Question number 7. Why are marine sticklebacks losing their pelvic spines when they started growing in a freshwater habitat? This is a well discussed example mentioned in the text as well as discussed by Professor Subramaniam. When marine sticklebacks started growing in a freshwater habitat, why did they lose their pelvic spines? And the options are: there was no consequence of having or losing the pelvic spine. Option B: pelvic spines that were useful in marine environments against predators were no longer necessary, hence they just lost it. Option C: in freshwater habitat, parasites would lodge themselves onto the pelvic spines, and hence sticklebacks without the pelvic spines survive better in this environment. Option D: in freshwater habitats, though pelvic spines help protect against predators, they decrease survival fitness. Hence, they lost the pelvic spines. So these are four explanatory statements. You have to tell me which is the correct reason why marine sticklebacks are losing their pelvic spines. Let's take some time to collect some answers. If you remember the example that we discussed about marine sticklebacks and their pelvic spines, this is an example of a particular modularity in a gene expression where the species has adapted to its new environment while losing its pelvic spine. Okay, it's time for me to reveal the correct answer. The correct answer to this question is option C. In freshwater habitat, parasites would lodge themselves onto the pelvic spines, and hence sticklebacks without the pelvic spine survive better in this environment. So in salt water or in marine habitat, when the sticklebacks had pelvic spines, there were no parasites to lodge into them, but when they encountered a new habitat in the freshwater, there were dragonfly parasites which would lodge onto these pelvic spines, causing a hindrance to the survivability of these sticklebacks. That's why across evolution, those sticklebacks which are now freshwater lose their pelvic spine. So we have two forms of sticklebacks now. Stickleback, sticklebacks are fishes which are both marine and freshwater origin. Ocean going form which have long spines protects fish against fish predators. So we have different selection pressure for the same organism in different habitats. In marine environment, big fishes are the predators which decrease the survivability of sticklebacks. So to protect against predation of big fishes, these sticklebacks have long spines, pelvic spines. While those which have now moved on to the freshwater habitats or in the lake bottoms, these fishes have lost the pelvic spine to decrease the dragonfly larva predation. So there are certain parasitic larvae called the dragonfly larva which lodge into these pelvic spines and predate and eat up the fish from the within. So is uh, this is also known as devolution, the term coined, which is backwards evolution, in which the freshwater stickleback has a corrupted genetic switch that stops expression of PIT-X1 gene in the pelvic region, preventing spine formation in that area. So PIT-X1 gene is the one responsible for production of the pelvic spine. The ocean going form and the as well as the lake dwelling form both have the PIT-X1 gene, but there is a genetic switch or a modularity in one of the enhancers, hind limb specific enhancer, where uh, the enhancer now represses and transcription factor cannot bind PITX1, thereby suppressing the formation of pelvic spine in that area. This helps avoid dragonfly larva predation because the larva attach to the spines, but it is the wrong type of change required to climb Mount Improbable. So another regional advantage. So let's ignore that. The main advantage this freshwater sticklebacks gain from losing their pelvic spine is the attack from dragonfly larvae. So these larvae can lodge onto the pelvic spine and then attack the fish, whereas in the marine form, big fish which predate on these sticklebacks, the pelvic spine helps guard them against the big fishes. So evolutionarily, the genetic switch that stops the expression of the spit X1 gene responsible for development of stickleback spine. Okay, moving on to the next question. If Plasmodium vivax infects by binding to Duffy, which is an essential protein, how can some individuals develop resistance to Plasmodium vivax? So this is a question related to malarial infection and the Duffy coat protein. Again, if I hint it, it's an example of enhancer modularity. Options are: if there are mutations in the Duffy's coding region such that Plasmodium cannot bind to it, since Duffy has an essential role in other parts of the body, the individual will not develop properly. Option B: mutation in Duffy's enhancer allow modification of its expression only on RBCs and retain its critical functions elsewhere. Option C: mutations in Duffy's enhancer region are lethal to the organism as it is an essential protein. And option D: Plasmodium vivax developed a mutation that could no longer bind to Duffy. These are the four options, and you have to tell me which is the correct answer. If Plasmodium vivax infects by binding to Duffy, which is an essential protein, how can some individuals develop resistance to Plasmodium vivax? So I'll wait for a minute and then I'll reveal the correct answer. Okay, it's time for me to reveal the correct answer. I see Ajay has answered option Option 2, mutations in Duffy's enhancer allow modification of its expression only on RBCs and retain its critical functions elsewhere. And this is the correct answer. Ajay has again answered the correct answer. This is the reason why Plasmodium vivax resistant population exists, because such individuals have certain mutations in enhancer of the Duffy protein, enhancer which regulates the Duffy protein, leading to modification of its expression only on the RBCs and retain its critical function elsewhere. So these mutations in the enhancer only modifies the expression on the RBCs, and since we know that RBCs are the target for Plasmodium vivax parasite, these parasites lodge into the RBCs and then develop into the sporozoid stage and infect the individual and other propagate their infection. So since now the Duffy protein is modified on the RBC expression, now the Plasmodium vivax cannot infect the RBC, and such individuals develop resistance to Plasmodium vivax. So if we have here is a representation of the Duffy antigen on the RBC and other such antigens, and on the cognate side we have Plasmodium vivax with surface proteins, the Duffy binding protein, which can recognize these Duffy antigen receptors, and that's how the Plasmodium vivax latches onto the RBC using its Duffy binding protein, recognizing the Duffy antigen, the Duffy cognate receptor present on the RBCs. So now many therapies and strategies to reduce malarial infection produce such antibodies which can neutralize the Duffy binding protein or the reticulocyte binding protein, uh, preventing the latching of Plasmodium vivax onto RBC surfaces. So Duffy antigen is an important protein which allows the infection of Plasmodium vivax onto RBCs. Now what happens when what happens to individual with a mutation in Duffy enhancers? So let's revisit again. Plasmodium vivax is one of the species of protozoan that causes malaria. Some African populations are immune to P. vivax because their RBCs lack the Duffy glycoprotein that the P. vivax uses to attach to the RBC. So such populations do exist uh, in South African populations. Some of the African populations are immune because they have no Duffy protein on their RBCs. Duffy glycoprotein is a receptor for IL-8 and is also found on cerebellar Purkinje neurons, blood and veins. So these are the other regions where Duffy glycoprotein is used. So this shows the modularity of the Duffy enhancer. So the same set of gene can be expressed in different tissues at different time point, owing to its developmental origin and the requirement in as well as in RBCs, Purkinje neurons, blood veins. People people immune to P. vivax express Duffy on Purkinje neurons and blood vessels, but not in the RBCs. So mind you that only only on RBCs does there is a change in Duffy expression, but not in other tissues such as Purkinje neurons and blood vessels. A mutation in the RBC specific enhancer for Duffy has allowed these people to retain expression of Duffy in the other cells while losing it in the RBC. So here is the enhancer modularity reason. Only the RBC specific enhancer harbors a mutation which does not produce Duffy coat only in the RBC, but the Purkinje blood vessels and every other cell type which naturally expresses Duffy coat is still expressing it. Thus, the modular nature of the Duffy enhancer has made it possible for these population to be resistant to P. vivax infection. So here is the reason that we found out and we have answered correctly. So this is a best example of enhancer modularity, where the modularity of enhancer in the Duffy gene expression only modifies it in the RBCs, leading to immunity against P. vivax. I hope it's clear to the students. Great, cool. Moving on to the next question. Question number 9. Which of the following are true? It's a true false type state question about Hox genes. Hox genes specify anterior posterior axis only in mammals. Option B. Hox genes specify anterior posterior axis in a wide variety of organisms, and this shows that these genes arose from a common ancestor. Option C. Hox genes of insects and humans are paralogs. Option D. Hox genes of insects and humans are orthologs. So these are the four options. You have to identify the statements which are true. Mind you that there can be multiple statements, multiple choice answers. There can be more than one statement true. Hox genes specify anterior posterior axis only in mammals. Hox genes specify anterior posterior axis in a wide variety of organisms, and this shows that these genes arose from a common ancestor. Hox genes of insects and humans are paralogs. Hox genes of insects and humans are orthologs. Okay, I see Ajay has answered option 2 and option 3. Okay, now let me reveal what are the correct answer to this question. The first correct answer to this question is option 2. Hox genes specify the anterior posterior axis in a wide variety of organisms, and this shows that these genes arose from a common ancestor. So option 2, this statement is true. And the next statement which is true is option 4. Hox genes of insects and humans are orthologs. They are not paralogs but orthologs. I think Ajay is partially correct. So correct answer would be option 2 and 4. Orthologs are genes with similar property in different species, while paralogs are different genes with different properties in same species. So paralogs occur due to duplication and divergence events, while orthologs occur due to speciation events. So here is a schematic representation showing the different Hox genes and their involvement in patterning of not only mammals but in Drosophila, in Cnidarians, in Annelids, and in every other order, which shows that these genes share common ancestry. So in all bilaterally symmetrical animals, the anterior to posterior axis is defined by the Hox genes, which includes all classes of animals like from vertebrates to cnidarians to flatworms to insects, and there are many Hox genes here. In cnidarias we see them NVAX6 specifying the oral to aboral axis, NVAX1 from the aboral, annelids anterior, central and posterior axis, arthropoda or in Drosophila melanogaster heavily studied anterior to posterior axis with the Hox genes, and humans. So along the neural tube and as well as the vertebral column, we have the expression of different Hox genes specifying the vertebra, which we just encountered in an earlier question: the cervical, thoracic, lumbar, caudal, and sacral. So all these anterior to posterior patterning axes are defined by Hox gene expression. And again, I would like to emphasize the subtle difference between what is an ortholog, what is an paralog, what is an analog, what is a homolog. So if we have gene A here, just go with me. There is a gene duplication event and there is a speciation event, which now one of the copies is still gene A, but the other copy has become gene B. Now gene A undergoes speciation. Gene A now occurs in two different species, the red branching that we see here, gene A1 and A2. So gene A1 and A2 are orthologs, since the same gene A has now undergone speciation into two different species, A1 and A2, performing mostly similar function. So pro genes or proteins in two different species performing similar function, sharing common ancestry, is the ortholog. While if we compare gene A1 and B1, B is the one which arose from gene duplication and divergence in the first place. If we compare gene A1 and B1 present in the same species, species 1, then those will be known as paralogs. So two genes or proteins in the same species performing different functions arose due to gene duplication and divergence is a paralog. And gene C is an analog because it is totally unrelated. So both orthologs and paralogs are homologs, means they share common ancestry, but paralogs are within species and may or may not have different functions. Orthologs are between two different species which which generally have similar functions. So if we see such patterning orthologs, uh, the orthologs between fly and human. So the question was, Hox genes of insects and humans are orthologs. This was the correct answer. So since we have two different species here, Drosophila melanogaster belongs to insects and human belongs to the mammals. So between insects and mammals, we have the same Hox genes performing similar functions, the Hox genes. Thereby, they will be known as orthologs. So the correct answer is option 2 and option 4. Option 2, because Hox genes are involved in anterior posterior patterning of many organisms, hence they share a common ancestry. And Hox genes of insects and humans are orthologs since they are two different species. I hope this is clear to the students. Good, great. Moving on to the last question of this session. Which of the following are false? Again, a true false type question. Which of the following are false? Option A. In deep homology, not just individual genes, but entire pathways are conserved. Option B. Deep homology supports that certain features like nervous system may have developed independently in different species. Option C. Accumulation of small genetic changes are sufficient to create entirely new features. And option D. More than mutations in the protein coding region, mutations in the regions that define when, where, and how long a protein will be expressed lead to formation of entirely new features. So you have to identify which of these statements are false. Mind you, there might be more than one correct option. So the first two statements are about deep homology, which talks about the development of nervous system and conservation of pathways, and the next two statements are regarding variations in the evolutionary time scale in the fixed developmental regulation, accumulation of small genetic changes, are they sufficient to create entirely new features, and where, when, where, and how long a protein will be expressed, mutation in those regions will define the formation of entirely new features. So let's wait and collect some answers. Yes, Ajay, homolog is a broader group. Homolog both consist of paralogs and orthologs. Paralogs within the same species, orthologs within different species. Paralogs may or may not have different functions, but generally they have do have different functions. Orthologs similar majorly have same functions, because they occurred via a speciation event. So if you understand what is speciation, means at a time point, one ancestor gave rise to uh, two different organisms. So the gene that was present in the ancestor is just copied into the two different organisms. Now those two different organisms are having the same kind of gene. Yes, Ajay wanted to. Yeah, yeah. Analog is, yes. It's actually, it's, yeah, within, yes, yes. Yeah, orthologs and homologs are orthologs and paralogs are subset of homologs. You can say that within species and across species. Yes, yes, yes. You are correct. Yeah, great. Okay, to answer the 10th question that Ajay has mentioned, option 1 and 4. I think you might have to reconsider your options because read the statements. Which of the following are false? Are you sure? In deep homologies, not just individual genes but entire pathways are conserved. And fourth option, more than mutations in protein coding region, mutations in the region that define when, where, and how long a protein will be expressed lead to formation of entirely new features. Yes, see now that you change your answers, option 2 and 3, and you are very correct. Options 2 and 3 are the correct answers. These are the two false statements. Deep homology supports that certain features like nervous system may have developed independently in different species. This is a false statement. Deep homology, on the contrary, validates the converse of this, that the same nervous system in many animals is sharing deep homology, sharing a similar kind of developmental plan across different species, and accumulation of small genetic changes are not sufficient to create entirely new features uh, like teeth and neural crest cells. This is what we also encountered in the NPTEL lectures by Professor Subramanian. Okay, let us quickly go about what is deep homology. So here I have used the same example to explain you guys. In some instances, homologous pathways made of homologous components are used for same function in protostomes and deuterostomes. Protostomes and deuterostomes, this has been called deep homology. So here we have a Drosophila and a mouse. Drosophila is a protostome, mouse is a deuterostome. The same set of instructions form the nervous system in mice and flies. Here is an example, the nervous system example. Same set of instruction from the nervous system in mice and fly. Here we have a protein called DPP, which have homologous protein in mouse called BMP. BMP and DPP share homology. DPP in Drosophila, BMP in mouse. Cordin is the Cordin, on the other hand, specifies the inhibitor of BMP, whereas we have SOG as the inhibitor of DPP here in Drosophila. In the fruit fly, the TGF beta family member DPP is expressed dorsally and opposed by SOG ventrally. So dorsal expression of DPP in Drosophila, and in the ventral side we have SOG, which opposes the DPP, inhibitor of DPP. In the mouse, the TGF beta family member BMP4 is expressed ventrally and countered by the cordine dorsally. So we may have a difference in the change in the expression, but similar functions is observed. So here also we have an activator and an inhibitor specifying two different regions of the body axis in the animal. The highest concentration of cordine becomes the midline, which is dorsal in vertebrates and ventral in invertebrates. So we have opposite kind of body plan here, but the highest amount of cordine will become the midline, which is dorsal to the vertebrates or deuterostomes like mouse, and ventral to the Drosophila, which is a protostome. So we have ventral cord here, nerve cord. We have dorsal nerve cord, notochord, and the neural tube are dorsal structures in the mouse, while they are ventral structures in the Drosophila. But they are specified by the similar signaling program wherever there is highest coordin expression of SOG expression. Those are both inhibitors of DPP and BMP4 respectively. So the highest concentration of coordin becomes a midline, which is dorsal in vertebrates and ventral in invertebrates. The concentration gradient of BMP4, DPP4 then specify regions of the nervous system in the same order: MSH, MSX, IND, GSX, VND, and KX2. So highest concentration of inhibitor at the dorsal side and in the ventral side respectively. Then we have different gradient cells which are experiencing different gradient of the BMP protein or the DPP protein, and the cell fate is determined in a similar manner both in the protostomes as well as in the deuterostomes, where in the Drosophila and mouse. So the first highest concentration of BMP will express MSX here, which is homologous to MSH. The next set of cells will be expressing GSX and NKX2. We have IND and VND, which are similar homologs found in mouse and Drosophila. So this is termed as deep homology. So deep homology says that certain features like nervous system has the same developmental plan across different species. So here we have BMP and its inhibitor, there we had DPP and its inhibitor, specifying different regions of the nervous system. And for the second option, mechanisms of evolutionary change, here I am quoting some scientists. In 1940s, Richard Goldsmith wrote that accumulation of small genetic changes was not sufficient to generate evolutionary novel structures such as neural crest, turtle shells, or feathers. He argued that such evolution could only occur through inheritable changes in genes that regulate development. So, some scientists found out that small changes accumulated over time cannot suddenly lead to emergence of a new structure, but not the changes in the gene itself, but the regulatory regions that change lead to formation of such structures. In 1977, this idea was extended by Francois Jacob. He said that evolution works with what it has by combining existing parts in new ways rather than creating new parts. So it doesn't create new parts altogether, but combines different existing body plans. So again, coming back to the molecular parsimony, we have the same set of toolkit. Uh, we have the same set of toolkit for developing all the body organs and new parts as well as evolution. So what does evolution does? Such tinkering would occur primarily in genes that construct the embryo and not in the genes that function in the adult. So genes which are involved in regulation of the embryonic development, such tinkering, combining the existing parts in new ways can create uh, new new structures, novel structures such as all these mentioned in neural crest and turtle shells. And Arthur argued that following four ways in which tinkering can occur at the level of gene expression to generate phenotypic variation available for natural selection. Heterotopy, change in location. Change in location of expression of a particular gene can lead to formation of a different structure. Heterochrony, change in time. The time of expression of a gene in the developmental time period, time of expression of gene is very important, and change in time can lead to development of different structures. Heterometry, change in amount of gene expression. So any phenomena which is altering the amount of gene expression in a particular organism can lead to formation of different structures, as we have seen in the presence of morphogen. Highest gradient of highest receiver, the morphogen gradient turns into distinct structure as compared to intermediate as well as lowest uh, receiver of the morphogen. Heterotypy, change in kind, the kind of protein or the kind of gene expression. If that is changed, then that can also bring about evolutionary change or evolution of a new structure. So to summarize, accumulation of small genetic changes is not sufficient to form evolutionary novel structures. Changes and mutation in the regulatory regions of protein coding genes, which decide when, where, and how long a gene expression should be turned on, actually lead to formation of new structures. So those regulatory regions, those modular regions, if get combined in different ways, can produce different level of gene expression in location, time, and amount and kind, leading to formation of evolutionarily novel structures. So that's why option 2 and 3 are the false statements, while 1 and 4 are the correct statements. Good, very good. With this, we come to the end of this problem solving session. Let me just stop. Yes, yeah, Ajay, you're clear enough. Monday sessions. Oh, you might have to ask Mr. Parth because I have no contact. I have no contact with him seriously. I think yeah. I think yeah, yeah, that might be a reason. And actually, in the first few classes, we had 10 to 12 people attending the classes, but then people just didn't come and they dropped off. I think the timing was not right for them, or maybe they found. Yeah, yeah, yeah. I can understand. Yeah, yeah, yeah. It's fine, Ajay. See, I have to do my job, so even if yeah, yeah, yes, yeah, that is why we see a even lesser population attending the live TA sessions. So in the first class, I had some 10 to 12 students attending, then but gradually it decreased. It's okay, maybe people can find these recordings and watch it over, but still I have to finish my job on my end so that I leave no stone unturned and students can actually derive help from these practice problem sessions whenever they want to view this. So it's good that you were here and we had such fruitful discussions. One more week of practice problem session to go, then this headache will be done. It's easier for the students since they are not attending these classes. But yeah, it's yeah. Thank you, Ajay. Okay, with that, I'll end this session and see you soon on the next Saturday. That will be the last class for this problem solving session, where we'll solve week 12 lectures for Introduction to Developmental Biology. And good luck for your exams. Thank you.